Chapter 1. Sexual Reproduction in Flowering Plants. Are we not lucky that plants reproduce sexually? The myriads of flowers that we enjoy gazing at, the scents and the perfumes that we swoon over, the rich colors that attract us are all there as an aid to sexual reproduction. Flowers do not exist only for us to be used for our own selfishness. All flowering plants show sexual reproduction. A look at the diversity of structures of inflorescences, flowers and floral parts shows an amazing range of adaptations to ensure formation of the end products of sexual reproduction, the fruits and seeds. In this chapter, let us understand the morphology, structure, and the process of sexual reproduction in flowering plants. Angiosperms. Flowers, a fascinating organ of angiosperms. Human beings have had an intimate relationship with flowers since time immemorial. Flowers are objects of aesthetic, ornamental, social, religious, and cultural values. They have always been used as symbols for conveying important human feelings such as love, affection, happiness, grief, mourning, etc. List at least 5 flowers of ornamental value that are commonly cultivated as in homes and in gardens. Find out the names of 5 more flowers that are used in social and cultural celebrations in our family. Have you heard of floriculture? What does it refer to? To a biologist, flowers are morphological and embryological marvels and sites of sexual reproduction. In earlier classes, we have read the various parts of a flower. Figure 1.1 will help you recall the parts of a typical flower. Can you name the 2 parts in a flower in which the 2 most important units of sexual reproduction develop? Pre-fertilization, structure and events. Much before the actual flower is seen on a plant, the decision that the plant is going to flower has been taken. Several hormonal and structural changes are initiated which lead to the differentiation and further development of floral primordium. Inflorescences are formed which bear the floral buds and then the flowers. In the flower, the male and the female reproductive structures, the androecium and the gynoecium, differentiate and develop. You would recollect that the androecium consists of a whole of stamens representing the male reproductive organ and the gynoecium represents the female reproductive organ. Stamen, microsporangium and pollen grains. Figure 1.2a shows the 2 parts of a typical stamen. The long and slender stalk is called the filament and the terminal generally bilobed structure is called anther. The proximal end of the filament is attached to a thalamus or the petal of a flower. The number and the length of stamens are variable in flowers of different species. If you were to collect stamen each from 10 flowers, each from different species, and arrange them on a slide, you would be able to appreciate the large variation in size seen in nature. Careful observation of each stamen under a dissecting microscope and making neat diagrams would elucidate the range in shape and attachment of anthers in different flowers. A typical angiosperm anther is bilobed, with each lobe having 2 theca. That is, they are diathious. Figure 1.2b. Often a longitudinal groove runs lengthwise separating the theca. Let us understand the various types of tissues and their organization in the transverse section of an anther. The bilobed nature of an anther is very distinct in the transverse section of the anther. The anther is a 4-sided tetragonal structure consisting of 4 microsporangia located at the corners, 2 in each lobe. The microsporangia develop further and become pollen sacs. They extend longitudinally all through the length of an anther and are packed with pollen grains. Structure of microsporangium. In a transverse section, a typical microsporangium appears near circular in outline. It is generally surrounded by 4 wall layers, figure 1.3b, the epidermis, endothecium, middle layers and tapetum. The outer 3 layers perform the function of protection and help in dehiscence of anther to release the pollen. The innermost layer, the tapetum, it nourishes the developing pollen grains. Cells of the tapetum possess dense cytoplasm and generally have more than one nucleus. Can you think of how tapetal cells could become binucleate? When the anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissue occupies the center of each microsporangium. Microsporogenesis. As the anther develops, the cells of the sporogenous tissue undergo meiotic divisions to form microspore tetrads. What would be the ploidy of the cells of the tetrad? As each cell of the sporogenous tissue is capable of giving rise to microspore tetrad, each one is a potential pollen or microspore mother cell. The process of formation of microspores from a pollen mother cell PMC through meiosis is called microsporogenesis. The microspores, as they are formed, are arranged in a cluster of 4 cells, the microspore tetrad, figure 1.3a. As the anthers mature and dehydrate, the microspores dissociate from each other and develop into pollen grains, figure 1.3b. Inside each microsporangium, several thousands of microspores or pollen grains are formed that are released with the dehiscence of anther, figure 1.3c. Pollen grain. The pollen grains represent the male gametophytes. If you touch the opened anthers of hibiscus or any other flower, you would find deposition of yellowish powdery pollen grains on your fingers. Sprinkle these grains on a drop of water taken on a glass side and observe under microscope. You will really be amazed at the variety of architecture, size, shape, color, design seen on the pollen grains from different species. Figure 1.4. Pollen grains are generally spherical, measuring about 25 to 50 micrometers in diameter. It has a prominent two-layered wall. The hard outer layer called the exine is made up of sporopollenin, which is one of the most resistant organic matter known. It can withstand high temperatures and strong acids and alkali. No enzyme that degrades sporopollenin is so far known. Pollen grain exine has prominent apertures called germ pores, where sporopollenin is absent. Pollen grains are well preserved as fossils because of the presence of sporopollenin. The exine exhibits a fascinating array of patterns and designs. Why do you think the exine should be hard? What is the function of germ pore? The inner wall of the pollen grain is called the intine. It is a thin and continuous layer made up of cellulose and pectin. The cytoplasm of pollen grain is surrounded by a plasma membrane. When the pollen grain is mature, it contains 2 cells, the vegetative cell and the generative cell, figure 1.5b. The vegetative cell is bigger, has abundant food reserve and a large, irregularly shaped nucleus. The generative cell is small and floats in the cytoplasm of the vegetative cell. It is spindle-shaped with dense cytoplasm and a nucleus. In over 60% of angiosperms, pollen grains are shed at the 2-cell stage. In the remaining species, the generative cell divides mitotically to give rise to the 2 male gametes before pollen grains are shed in 3-cell stage. Pollen grains of many species cause severe allergies and bronchial afflictions in some people, often leading to chronic respiratory disorders like asthma, bronchitis, etc. It may be mentioned that Parthenium or carrot grass that came into India as a contaminant with imported wheat has become ubiquitous in occurrence and causes pollen allergy. Pollen grains are rich in nutrients. It has become a fashion in recent years to use pollen tablets as food supplements. In Western countries, a large number of pollen products in the form of tablets and syrups are available in the market. Pollen consumption has been claimed to increase the performance of athletes and racehorses. When once they are shed, pollen grains have to land on the stigma before they lose viability, if they have to bring about fertilization. How long do you think the pollen grains retain viability? The period for which pollen grains retain viability is highly variable and to some extent depends on the prevailing temperature and humidity. In some cereals such as rice, wheat, pollen grains lose viability within 30 minutes of their release, and in some members of Rosaceae, Leguminosae, and Solanaceae, they maintain viability for months. You may have heard of storing semen or sperms of many animals, including humans, for artificial insemination. It is possible to store pollen grains of a large number of species for years in liquid nitrogen, minus 196 degrees centigrade. Such stored pollens can be used as pollen banks, similar to seed banks in crop breeding programs. The pistil, megasporangium or ovule, and embryo sac. The gynoecium represents the female reproductive part of the flower. The gynoecium may consist of a single pistil, monocarpellary, or may have more than one pistil, multicarpellary. When there are more than one, the pistils may be fused together, syncarpous, figure 1.7b, or may be free, apocarpous, figure 1.7c. Each pistil has three parts: the stigma, style, and the ovary. The stigma serves as a landing platform for pollen grains. The style is the elongated slender part beneath the stigma. The basal bulged part of a pistil is the ovary. Inside the ovary is the ovarian cavity or locule. The placenta is located inside the ovarian cavity. Recall the definition and the types of placentation that you have studied in class 11. Arising from the placenta are the megasporangia, commonly called ovules. The number of ovules in an ovary may be 1, wheat, paddy, mango, to many, as in papaya, watermelon, and orchids. The megasporangium or ovule. Let us familiarize ourselves with the structure of a typical angiosperm ovule. The ovule is a small structure attached to the placenta by means of a stalk called funicle. The body of the ovule fuses with the funicle in the region called hilum. Thus, hilum represents a junction between ovule and funicle. Each ovule has one or two protective envelopes called integuments. Integuments encircle the nucellus except at the tip, where a small opening called the micropyle is organized. Opposite to the micropylar end is the chalaza, representing the basal part of the ovule. Enclosed within the integuments is a mass of cells called the nucellus. Cells of the nucellus have abundant reserve food materials. Located in the nucellus is the embryo sac or female gametophyte. An ovule generally has a single embryo sac formed from a megaspore. Megasporogenesis. The process of formation of megaspores from the megaspore mother cell is called megasporogenesis. Ovules generally differentiate a single megaspore mother cell or MMC in the micropylar region of the nucellus. It is a large cell containing dense cytoplasm and a prominent nucleus. The MMC undergoes meiotic division. What is the importance of MMC undergoing meiosis? Meiosis results in production of 4 megaspores. Female gametophyte. In a majority of flowering plants, one of the megaspores is functional while the other 3 degenerate. Only the functional megaspore develops into female gametophyte or embryo sac. This method of embryo sac formation from a single megaspore is termed as monosporic development. What will be the ploidy of the cells of the nucellus, MMC, and functional megaspore and female gametophyte? Let us study about the formation of embryo sac in detail. Figure 1.8b. The nucleus of the functional megaspore divides mitotically to form 2 nuclei which move to the opposite poles, forming the 2-nucleate embryo sac. 2 more sequential mitotic nuclear divisions result in the formation of 4 nucleate and later 8 nucleate stages of the embryo sac. It is of interest to note that these mitotic divisions are strictly free nuclear, that is, nuclear divisions are not followed immediately by cell wall formation. After the 8 nucleate stage, cell walls are laid down, leading to the organization of a typical female gametophyte or embryo sac. Observe the distribution of cells inside the embryo sac in figure 1.8b and c. 6 of the 8 nuclei are surrounded by cell walls and organized into cells. The remaining two nuclei, called polar nuclei, are situated below the egg apparatus in the large central wall cell. There is a characteristic distribution of the cells within the embryo sac. Three cells are grouped together at the micropylar end and constitute the egg apparatus. The egg apparatus in turn consists of two synergids and one egg cell. The synergids have special cellular thickenings at the micropylar tip called filiform apparatus, which play an important role in guiding the pollen tube into the synergid. Three cells are at the chalazal end and are called antipodals. The large central cell, as mentioned earlier, has two polar nuclei. Thus, a typical angiosperm embryo sac at maturity, though 8-nucleate, is 7-celled. Pollination. In the preceding sections, you have learned that the male and female gametes in flowering plants are produced in pollen grains and embryo sac, respectively. As both types of gametes are non-motile, they have to be brought together for fertilization to occur. How is this achieved? Pollination is the mechanism to achieve this objective. Transfer of pollen grains shed from the anther to the stigma of a pistil is termed as pollination. Flowering plants have evolved an amazing array of adaptations to achieve pollination. They make sure of external agents to achieve pollination. Can you list the possible external agents? Kinds of pollination. Depending on the source of pollen, pollination can be divided into three parts. Autogamy. In this type, pollination is achieved within the same flower. Transfer of pollen grains from the anther to the stigma of the same flower. In a normal flower, which opens and exposes the anthers and the stigma, complete autogamy is rather rare. Autogamy in such flowers requires synchrony in pollen release and stigma receptivity, and also their anthers and stigma should be close to each other so that self-pollination can occur. Some plants such as viola, commonly pansy, oxalis and commelina, produce two types of flowers: chasmogamous flowers, which are similar flowers of other species with exposed anthers and stigma, and cleistogamous flowers, which do not open at all. In such flowers, the anthers and stigma lie close to each other. When anthers dehisce in the flower buds, pollen grains come in contact with the stigma to effect pollination. Thus, cleistogamous flowers are invariably autogamous, as there is no chance of cross-pollen landing on the stigma. Cleistogamous flowers produce a sure seed set even in the absence of pollinators. Do you think that cleistogamy is advantageous or disadvantageous to the plant? Why? Gaetogamy. Transfer of pollen grains from the anther to the stigma of another flower of the same plant. Although gaetogamy is functionally cross-pollination involving the pollinating agent, genetically it is similar to autogamy, since the pollen grains come from the same plant. Xenogamy. Transfer of pollen grains from anther to the stigma of a different plant. Figure 1.9b. In this is the only type of pollination which during pollination brings genetically different types of pollen grains to the stigma. Agents of pollination. Plants use two abiotic, wind and water, and one biotic, animals agents, to achieve pollination. Majority of plants use biotic agents for pollination. Only a small proportion of plants use abiotic agents. Pollen grains coming in contact with the stigma is a chance factor in both wind and water pollination. To compensate for these uncertainties and associated loss of pollen grain, flowers produce enormous amount of pollen when compared to the number of ovules available for pollination. Pollination by wind is more common among abiotic pollinations. Wind pollination also requires that the pollen grains are light and non-sticky so that they can be transported in wind currents. They often possess well-exposed stamens so that the pollens are easily dispersed into the wind currents, and large, often feathery stigma to easily trap airborne pollen grains. Wind-pollinated flowers often have a single ovule in each ovary and numerous flowers packed into an inflorescence. A familiar example is the corn cob, The tassels you see are nothing but the stigma and style, which wave in the wind to trap pollen grains. Wind pollination is quite common in grasses. Pollination by water is quite rare in flowering plants and is limited to about 30 genera, most commonly monocotyledons. As against this, you will recall that water is a regular mode of transport for the male gametes among the lower plant groups such as algae, bryophytes and pteridophytes. It is believed, particularly for some bryophytes and pteridophytes, that their distribution is limited because of the need for water for the transport of male gametes and fertilization. Some examples of water-pollinated plants are Vallisneria and Hydrilla, which grow in fresh water, and several marine sea grasses such as Zostera. Not all aquatic plants use water for pollination. In a majority of aquatic plants such as water hyacinth and water lily, the flowers emerge above the level of water and are pollinated by insects or wind, as in most of the land plants. In Vallisneria, the female flower reach the surface of water by long stalk, and the male flowers or pollen grains are released on the surface of water. They are carried passively by water currents. Some of them eventually reach the female flowers and the stigma. In another group of water-pollinated plants such as sea grasses, female flowers remain submerged in water and the pollen grains are released inside the water. Pollen grains in many such species are long, ribbon-like, and they are carried passively inside the water. Some of them reach the stigma and achieve pollination. In most of water-pollinated species, pollen grains are protected from wetting by a mucilaginous covering. Both wind and water-pollinated flowers are not very colorful and do not produce nectar. What would be the reason for this? Majority of flowering plants use range of animals as pollinating agents. Bees, butterflies, flies, beetles, wasps, ants, moths, birds, sunbirds and hummingbirds, and bats are the common pollinating agents. Among the animals, insects, particularly bees, are the dominant biotic pollinating agents. Even larger animals such as some primates, lemurs, arboreal, tree-dwelling rodents, or even reptiles, gecko lizard and garden lizard, have also been reported as pollinators in some species. Often flowers of animal-pollinated plants are specially adapted for a particular species of animal. Majority of insect-pollinated flowers are large, colorful, fragrant, and rich in nectar. When the flowers are small, a number of flowers are clustered into an inflorescence to make them conspicuous. Animals are attracted to flowers by color and or fragrance. The flower pollinated by flies and beetles secrete foul odors to attract these animals. To sustain animal visits, the flowers have to provide rewards to animals. Nectar and pollen grains are the usual floral rewards. For harvesting the rewards from the flower, the animal visitors come in contact with the anthers and the stigma. The body of the animal gets a coating of pollen grains, which are generally sticky in animal-pollinated flowers. When the animal carrying pollen on its body comes in contact with the stigma, it brings about pollination. In some species, floral rewards are in providing safe spaces to lay eggs. An example is that of the tallest flower of Amorphophallus. The flower itself is about 6 feet in height. A similar relationship exists between the species of moth and the plant yucca, where both species, moth and the plant, cannot complete their life cycles without each other. The moth deposits its eggs in the locule of the ovary, and the flower, in turn, gets pollinated by the moth. The larvae of the moth comes out of the eggs as the seed starts developing. Why don't you observe some flowers of the following plants or any other available to you? Cucumber, mango, peepal, coriander, papaya, onion, lobia, cotton, tobacco, rose, lemon, eucalyptus, banana. Try to find out which animals visit them and whether they could be pollinators. You will have to patiently observe the flowers over a few days and at different times of the day. You could also try to see whether there is any correlation in the characteristics of a flower to the animal that visits it. Carefully observe if any of the visitors come in contact with the anthers and the stigma, as only such visitors can bring about pollination. Many insects may consume pollen or the nectar without bringing about pollination. Such floral visitors are referred to as pollen or nectar robbers. You may or may not be able to identify the pollinators, but you will surely enjoy the efforts. Outbreeding devices. Majority of flowering plants produce hermaphrodite flowers, and pollen grains are likely to come in contact with the stigma of the same flower. Continued self-pollination results in inbreeding depression. Flowering plants have developed many devices to discourage self-pollination and to encourage cross-pollination. In some species, pollen release and stigma receptivity are not synchronized. Either the pollen is released before the stigma becomes receptive, or the stigma becomes receptive much before the release of pollen. In some other species, the anther and the stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower. Both the devices prevent autogamy. The third device to prevent inbreeding is self-incompatibility. This is a genetic mechanism and prevents self-pollen from the same flower or other flowers of the same plant from fertilizing the ovules by inhibiting pollen germination or pollen tube growth in the pistil. Another device to prevent self-pollination is the production of unisexual flowers. If both male and female flowers are present on the same plant, such as castor and maize monoecious, it prevents autogamy but not gaetogamy. In several species such as papaya, male and female flowers are present on different plants. That is, each plant is either male or female, dioicy. This condition prevents from both autogamy and gaetogamy.